Today I'll talk about the human immune system as a source of biomarkers, and specifically the use of autoantibodies as biomarkers. We know that in all chronic conditions that we've studied so far at Sengenics, including autoimmune diseases, cancers, and neurodegenerative diseases, the body raises an autoantibody response to the disease state. So I'll briefly introduce our technology and share with you some published studies. So we started about 13 years ago as a Cambridge University spin-out company using technology developed by our Chief Scientific Officer, Professor Jonathan Blackburn. Our protein production and high-throughput facility is located in Kuala Lumpur. Last year we received major investment from Summa Equity, which is allowing us to expand our operations and develop our Boston facility. We have a broad patent portfolio covering all our core technology, as well as some diagnostic and adverse drug response IP. There we go. We, of course, work with a wide range of organizations. We work with 9 of the top 10 pharmaceutical companies, as well as many others, and numerous academic groups. We've worked with several of the major disease foundations, winning a grant in 2020 from the Michael J. Fox Foundation to determine the role of autoantibodies in the progression of Parkinson's, and partnering with the ONJCRI to enhance safety of cancer immunotherapy drugs. Our CSO, Professor Jonathan Blackburn, sits on the Industrial Advisory Board of HUPO and on key steering committees for the FNIH Biomarker Consortium. So biomarkers are potentially really valuable tools to identify disease, to classify patients as responders or non-responders, and perhaps to stratify patients for clinical trials. We at Sengenics believe that the human immune system might be able to do a remarkable job of marking out aberrant disease-associated changes in proteins through the production of autoantibodies, and that these autoantibodies might provide a better class of biomarker that is more specific and more distinct to the disease state than DNA and protein markers have provided thus far. I don't propose to explain today how autoantibodies arise, but I will simply point out that autoantibodies should make good biomarkers because they're disease-specific, they manifest early in disease, they're stable, abundant, and easily accessible. I'd like to take a moment to note that antibodies and autoantibodies bind to folded protein structures, with the literature saying that roughly 90% of all B-cell epitopes are discontinuous, surface-exposed, and typically hydrophilic in nature. Anyone who's ever run a Western blot knows that antibodies can also bind non-specifically to exposed hydrophobic surfaces on unfolded proteins with very untidy results. So to identify physiologically relevant autoantibodies, you really need to work with correctly folded antigens, which, as I'm sure many of you know, isn't straightforward even across modest collections of proteins. And this is where our technology comes in. So we manufacture protein arrays coated with recombinant proteins, selected on the basis of their association with disease. The proteins are recombinant fusion proteins expressed in insect cells. The fusion moiety is biotin carboxyl carrier protein, BCCP, and it has a number of useful functions. It has a specific biotinylation site that is conformation dependent. So when this domain is correctly folded, it is biotinylated by endogenous biotin ligase. And this then allows the protein to attach to the streptavidin-coated array surface. This is shown here. So the color-driven is the protein of interest. In pale blue, we have BCCP, and in orange, we have biotin. However, misfolding of BCCP prevents biotinylation. In addition, BCCP acts as a folding sensor. Misfolding of the protein of interest causes co-translational misfolding of BCCP, and so these misfolded proteins do not attach to the array. Thus, biotinylation of BCCP indicates correct folding of the full-length protein. We've also spent some time in optimizing the surface chemistry of the arrays. The proteins are immobilized onto a proprietary streptavidin-coated hydrogel. This aqueous environment helps maintain protein integrity and prevents the proteins from drying out and denaturing. In many ways, the proteins behave as if they were in free solution. The single point of attachment between the biotin and the streptavidin allows for free rotation, and this allows for optimal presentation of the protein of interest to its ligand, in this case, autoantibodies. This then is the assay. In many ways, it's a highly miniaturized ELISA. We simply add sample to the array, and this can be from a variety of sources, most typically serum or plasma, but also CSF, lung lavage, urine, tissue homogenate, cell culture supernatants, and we have a client working on nasal swabs, and other non-blood sources such as nasal brushing or other mucosal surfaces should work as well. The autoantibodies bind to the arrayed antigens. We add a fluorescently labeled detection antibody, shown here as anti-IgG, but in fact it could be isotype or subclass specific. So it could be anti-IgG1, anti-IgG4, anti-IgA perhaps. And we then detect in a standard microarray scanner to get a readout. And this is a quantitative assay, not simply a binary yes-no answer. So we have a method for arraying correctly folded proteins, but what about functionality? We've developed functional assays for different classes of proteins. We have more than half the human kinome on the arrays, 336 different kinases. All of them have been shown to bind ATP. The receptor tyrosine kinases all autophosphorylate as expected, meaning that they're not only functional on the array, but they've also correctly homodimerized. And we can show that kinase inhibitors act as expected as well. We can look at DNA binding proteins. We've studied p53 and various mutants, though it would work for any transcription factor. And we can measure the same binding affinities as we see in solution. And we also know that it is present on the array in its tetrameric form. And we have physiological relevance to the hits that come out of our arrays, out of our assays. In autoimmune diseases, we typically find that known expected autoantigens light up in the assays. So here are some data from a recent SLE study with TROV2 and SSB lighting up strongly. And this provides a great deal of confidence that there is biological relevance to the hits that we come up with. The combination of correct folding and optimized surface chemistry means that we get great signal-to-noise rate, as well as linearity of detection and low limits of detection, typically less than 10 picograms per mil. And because we can fabricate thousands of replica arrays from a given batch of expressed protein, we also get great reproducibility in manufacturing, as well as in the downstream assays. So we have a number of standard off-the-shelf arrays. Immunome is our discovery platform with over 1,600 human proteins. The CTA array has more than 200 cancer testis antigens arrayed, and the p53 array has 100 wild-type and p53 mutants. Our ImmuSafe array is for SARS-CoV-2 applications. Finally, we can make bespoke custom arrays as required. And this, in fact, is typically how a project goes. That after the discovery phase, customers will specify a subgroup of proteins for their custom array. For the custom arrays, as well as specific proteins, we can array non-protein biomolecules and more complex targets, including virus-like particles and bacterial membrane preparations. So this then is our direction of travel. We have multiple chips with autoantigens arrayed. We can perform on-chip post-translational modifications, such as citrullination. We can assay for immunoglobulin isotypes and subclasses, and do dual-color assays. And we're moving towards on-chip effector function assays. So let me give you some specific examples of our technology in action. So this first case study is an SLE patient stratification study from Miles Lewis at Queen Mary's University London, published in 2018. So their study used 186 SLE patients and 188 matched healthy controls in the discovery group, and then followed up with 86 SLE patients and 90 matched healthy controls in the validation group. The discovery group identified 226 autoantibodies with p-values of less than 0.05, and the validation group clarified this down to 79 autoantibodies, which are shown in the volcano plot here. TROV2 and SSB-LAR are the top hits and are expected markers of SLE. Of these 79, in addition to TROV2 and SSB, 9 were previously reported to associate with SLE, giving 68 novel autoantigens. And of these 68, 5 were not known as autoantigens, but were from SLE susceptibility genes, giving a total of 14 hits identified on the immunome array that were previously reported to associate with SLE. So here they show an unsupervised hierarchical clustering, which identifies 4 groups. So we've got SLE1A, 1B, 2 in green, and 3 over here. And those groups are also replicated again in the validation cohort. And this was confirmed by cross-correlation and principal component analysis, as shown here. These autoantibody profiles stratify SLE patients and appear to correlate with disease phenotype and trajectories. So 2 key themes emerge, with cluster 2 shown in green, centered around SMAD2, SMAD5 signaling, and cluster 3 shown in blue, centered around TLR and FKB signaling. Intriguingly, this clustering hints at differing underlying pathogenic mechanisms and possibly at differing treatments for these SLE subgroups. Here they show the data in a different format. Each cluster of antigens was analyzed with string for interaction pathways, and this phylogenetic tree of autoantigens summarizing key protein functions was determined. From this, it is apparent that the different signatures address different underlying mechanisms. The authors point out that subgroup 3 in blue, so that's this one up here, might be susceptible to B-cell directed therapies, while subgroup 2, down here in green, might be targeted with non-B-cell therapies. And so this is likely to offer some direction for differential therapeutic decisions. So let me switch to diagnostic biomarkers for cancer. So early detection is hugely important in cancer care. Detecting primary melanoma tumors at an early stage results in a 5-year survival rate as high as 99%, whereas 5-year survival for late-stage patients is only 15 to 20%. Pauline Zenker notes that many blood-based biomarkers have been proposed, including microRNAs, circulating tumor cells, and circulating tumor DNA. However, none of these appear to be sufficiently sensitive to detect the initial transformation to malignancy and may therefore not be suitable diagnostic biomarkers for early-stage melanoma. They postulate that autoantibodies may provide a more advantageous blood-based biomarker as they reflect the initial humoral immune response against a tumor, and their increased levels can be detectable months to years prior to clinical evidence of a primary tumor. So in this study, they looked at stage 1, stage 2 melanoma patients. They had a large cohort of 124 patients. The initial study identified 139 biomarkers, including several previously associated with cancer and melanoma, some of which were shown up here. The top 139 identified biomarkers appeared primarily related to general cancer pathways, including apoptosis, pathways associated with the immune response and cell cycle, p53 signaling, and MAP kinase signaling pathway, the main pathway associated with melanomagenesis, highlighting the biological relevance of the identified biomarkers. Further analysis by machine learning identified a 10-panel marker with a combined sensitivity of 84%, specificity 79% for primary melanoma. It's important to note that the biomarkers included in the panel did not necessarily display the highest individual diagnostic potential. Instead, they're a combination, displaying a broader occurrence of positive seroreactivity for patient sera. Related to that, a separate study at the ONJ-CRI on stage 3, stage 4 melanoma patients identified a somewhat different marker set, but again with very high sensitivity and specificity, this time 93% and 89%. So now let me take a look at drug response prediction. So this study is from Chen's group at China Medical University Hospital, Taiwan, published earlier this year. And this comes on the tail of a smaller 2016 study by Hajiwara et al., showing that RA patients treated with infliximab can raise an immune response against this biologic, and that anti-Rho SSA antibodies associate with that response. The accumulating evidence indicates that the presence of anti-drug antibodies may be associated with low or undetectable drug levels and the ensuing reduction of therapeutic responsiveness to TNF-alpha inhibitors. This is shown here for adalimumab. The presence of anti-drug antibodies was determined by ELISA. As you can see here, antibody-positive patients have almost undetectable levels of adalimumab. And it should be noted that antibody-positive patients are also poor responders to adalimumab based on their EULAR score. So the study comprised 4 healthy controls plus 30 anti-drug antibody positive and 32 anti-drug antibody negative patients. There were no significant differences in the positive rate of rheumatoid factor or anti-citrulline peptide antibodies, dosage of corticosteroids or methotrexate between the antibody positive and antibody negative patients. Serum was collected and assayed on Sengenics immunoarray, and the resulting fluorescence gave rise to a panel of biomarkers associated with the presence of anti-drug antibodies. From the initial cohorts, we see here the top 20 putative biomarkers correlating with anti-drug antibody or poor EULAR response in plasma samples at baseline and week 24 of adalimumab therapy. The expression level of the differentially expressed autoantibodies shows quite clearly responder and non-responder groups. And at the bottom here you can see, so in purple we have the antibody negative patients, and in blue the good EULAR score patients, segregating quite nicely together. So the best subsets of markers were selected on a backward selection iterative process known as recursive feature elimination. The best panels were then stability tested by random forest analysis. Now, individual ROC curve analysis of the putative biomarkers revealed that anti-TROV2 antibody at baseline and at week 24 had a high discriminative power for predicting the emergence of anti-drug antibodies and poor EULAR response. However, when the biomarker panels were assessed, although TROV2 and SSB were found in all panels, the diagnostic performances, particularly of this panel of 8 biomarkers, was found to be superior. Finally, the authors note, we're the first to use a high-density protein microarray with machine learning approach to identify protein biomarkers that are predictive of anti-drug antibody development and therapeutic response to adalimumab. And it's worth reiterating here that this biomarker panel pre-existed in patients prior to their exposure to adalimumab. So I hope you found this presentation to be useful. I'd like to flag up a presentation in tomorrow morning's session by Herben Boomer from GSK, who will be presenting some data on a Sjogren's biomarker discovery program. It's a really powerful story, so I hope you get a chance to attend. So in summary, I hope I've shown that the Sengenics protein arrays are highly applicable to autoantibody profiling in many diseases. We've shown here and elsewhere their utility in early disease diagnosis, patient stratification, drug response, and prediction of adverse events. If you'd like to talk to us about how to apply our technology to your projects, you can find us at booth 39 for the next couple of days. And so with that, I'd like to thank you for your attention. I kept nicely to time, so thank you. The question master arrives.
Wow, shouldn't be late. Okay. Ask you guys if you have any questions. I don't know if anyone's been in the room with me this morning, but I've been trying to prompt people to, if they have any thoughts or comments, to come up to the microphone and add. Last session helped me out by 3 people coming up, so I don't know if there's anyone who wants to ask any questions or make any comments. If not, I will have to end the session because I don't want to keep you up here waiting. Thank you, so honestly. You're a star. There's twice today. Thank you.
Oh, my.
So I've noticed that you've mentioned that the platform that you use allows the proteins to have their own, the natural conformation, so to speak.
Yes.
Have you worked with proteins that have mobile things depending on the amount of calcium? I'm thinking some of the transglutaminases, for example, or do you have examples of proteins that are difficult to handle with the standard assays because their conformation change then makes them quite incapable of being detected by your standard antibody that only recognize the one conformation?
Yeah, that's a really good question. So I'm just trying to dredge through my memory here. So we have found generally that the proteins that we are looking for tend to be fairly well behaved. And so where, but we would, so sometimes for instance, for transmembrane proteins, we'll often, we'll eliminate the membrane portion and concentrate on either intracellular or extracellular, but we try to take the entirety of it. I can't think of an example where we have proteins that have are either quite mobile, have different, mobile domain structures, or largely disordered regions. Although I think the SARS-CoV-2 nuclear capsid has a domain which is quite disordered. I think that's right. And so we have that on the chip, and it seems to behave reasonably well. We pulled out a number of antibodies against that.
Right.
So I suspect it depends. I think that's what I'm trying to say.
Thank you.
Thank you.
Thank you so much for the question. Okay. Someone's also submitted a question via Slido, so thank you as well, Anonymous. Thank you for a really interesting talk. Where do you see this technology going in the future, and is it applicable to other drugs/diseases?
Yes. So we take the second part of the question first, I think. So we have evidence from a couple of other drug response or patient stratification diseases. So I think we have, as a company, we have fairly high confidence that the adalimumab study, for instance, would be applicable to a significant number of other drugs. Whether those would be only immunotherapies or whether it's more broadly applicable to therapeutics, I think we don't know the answer to that. But given that this is a pre-existing response, not a response to the drug itself, I suspect that it's a fairly widely applicable approach. Where we see the technology going, very much we would like to start driving towards IVD clinical testing. I think that there's a significant opportunity for early detection and for patient stratification, and also for assessing patients who either will or won't respond to particular drugs. And that's definitely the direction that we're leaning towards. So, thank you.
Thank you so much. We do have around about, if I check my watch again, I think it's about 5 minutes.
Okay.
Yeah, well, I lied, it was 6 minutes, but we're a minute off. Thank you so much. And we've had another question as well, but I will always let the person take priority, because I don't want you waiting.
Hi. Just coming from a clinical lab, how do you guys, you said there's about 1,600 proteins or antigens on your chip. How do you go about controlling that? And do you have any instances of false negatives, false positives, as in terms of like your biochip?
So for a moving towards a diagnostic or therapeutic test of some kind, we would so that would be part of the custom chip. So we would take our autoantibody signature, and we're looking at between 10 and 20 targets. Probably would be an appropriate number of autoantibodies to have a robust assay. In terms of so getting towards developing an IVD product, we know that our manufacturing process is robust enough to deliver the kind of reliability and reproducibility that you require. We're currently going through our first round of certification, so I don't know how well that's going to go. In all honesty, until it's done, we can never say it's done. But I think that's where we would go to. In terms of false positives, false negatives, we see a very low background of cross-reactivity. I don't know whether we've specifically looked or tried to force false negative or false positive tests on there. So I don't think we've artificially tried to force an error. So I couldn't answer that question, I'm afraid.
Thank you so much. And the question that's come through during that, well, before that, just to be clear, can you use the platform technology to create an antibody sandwich assay or just for direct ELISA style tests?
So the assay platform itself is fairly open. I'm not sure whether we would go down that route. So, but at the moment, and our preference is to be creating microarrays on this on a hydrophobic hydrogel-coated glass slide. That's not the only substrate that would be possible with our technology. So I potentially could see some kind of sandwich ELISA type thing. But I don't think we're going down that route. I don't think there's any work in the background that's going in that direction. If there proves to be a significant demand for such a thing, I think that we could do it, but I think that we're not going to at the moment.
Thank you so much. There's no additional questions on Slido, so I'll turn back to you, the audience. If anyone has any final comments or questions they would like to ask, now would, I'm happy for you to ask all the questions if you want. There's no problem here.
One of our issues that when we solve with our clinical assays, a lot of our patients were taking biotin supplements. I think there was a publication about patients supplementing their diet with biotin, and whether, have you guys tested that out? Does that biotin, high levels or any sort of level, does it actually interfere with your assay?
No. So obviously the array is the hydrogel is coated with streptavidin, but during the manufacturing process, after the proteins are spotted onto the array, the entire surface is blocked with biotin, and then subsequently there's a dried milk powder, a dried milk blocking step as well, which again milk has biotin. So no, we don't have. I don't think there would be any issues with that. Yeah, thank you.
Thank you so much. We have 2 more minutes left. Does anyone have any final questions or thoughts they would like to share? If not, I will close the session off. Don't see any movement, so yeah, I'm going to end the session there. Geoff, thank you so much for the presentation, and I'm sorry I almost scared you with being late.
Thank you very much, everybody. Thank you.